Hello world, as Tim Berners-Lee once famously typed at the start of the internet era. My name's Damien Bell. I am Global Director of Scientific Affairs at Sofian. We will be starting very shortly, so no waiting around. We've got two great speakers for you today talking about application of automated patch clamp in the characterization and screening of axoCells, which are these hiPSC-derived cardiomyocytes. We've got Dr. Jamie Bhagwan of AXOL, who will be starting off first. He'll be giving a 20-minute talk on these chamber specifics of ventricular versus atrial cardiomyocytes that they do these hiPSC derivations of. And afterwards, Kadla will present, Dr. Kadla Rosholm, my colleague at Sofian, will present on using these hiPSC cardiomyocyte cells in recordings, ion channel recordings on automated patch clamp platforms that Sofian are well known for. We will start shortly, but some admin before we do start. In the right-hand side of your screen, in most people's screens at least, you will have a chat tab. Please put any questions that you may have for the speakers or for our experts, because we will have a third expert joining Kadla and Jamie at the end when we answer all of your questions, or answer as many as we can do. So Dr. Atanaska Velikchova will be joining them as our culturing expert to discuss some of the potential questions around culturing of these cells. Those questions will come at the end, so we won't take questions at the end of each talk. We'll wait for both talks to finish. And then hopefully, with both talks being about 20 minutes long, we'll have still another 15 to 20 minutes remaining to answer as many of your questions that you can put in that chat panel on the right-hand side of your screen. On the right-hand side of the screen, we also have a polls tab. So intermittently through the talks, my colleague who is running things expertly in the background, Yetta will put up polls through the talks that hopefully you can answer whatever you fancy of those polls. The more the better, ideally, from our perspective. And finally, on that right-hand side panel, there are also a number of PDFs, so brochures, application reports from both AXOL and Sofian that are pertinent to these talks today. So, without further ado, I think we should get started so that we aren't taking too much of the speakers and the Q&A time. We will be having Jamie joining me on stage shortly now, and Jamie will be talking about chamber specificity of axoCells, hiPSC-derived cardiomyocytes, and metabolic maturation approaches. Over to you, Dr. Bhagwan.
Thank you very much, Damien, and thank you, Sofian, for inviting me to talk to you today about chamber-specific cardiomyocytes and some of the metabolic maturation approaches we're using at AXOL Bioscience. So I thought I'd start with a quick overview of AXOL Bioscience. So we're a company that manufactures iPSC-derived cells and provides services to support preclinical research and drug discovery. So we have over 70 patient-derived lines that we can differentiate to a number of cell types in the fields of neuroscience, ophthalmology, dermatology, and cardiovascular. But today, we're going to focus on the cardiomyocytes that we produce from iPSCs. So we have our off-the-shelf offering. We have a male and a female healthy donor that we can differentiate to both ventricular and atrial cardiomyocytes. So we provide these as frozen cryovials, either 1 million or 5 million cells per vial. And we also ship media and the ECM that's required to go along with culturing these cells. So our cells are, they'll spontaneously beat 2 to 3 days post-thaw, but we recommend that they're assay ready 7 days post-thaw. They express all the key cardiac markers you'd expect, so all the sarcomeric proteins. And when we look at our male and female donors, so when we compare our ventricular cardiomyocytes between the male and the female donor, we don't see any significant differences in electrophysiological characteristics when we look on the MEA, so conduction velocity, field potential duration, etc. However, we are aware of some emerging work looking at sex differences in hiPSC-derived cardiomyocytes. So it's definitely a new area of interest. But perhaps what AXOL has as a USP is our provision of both atrial and ventricular cardiomyocytes from the same donor. So every batch is QC'd, and we look at the ventricular and atrial isoforms of this myosin proteins, so MLC2B and MLC2A. And generally, we see that each chamber-specific cardiomyocyte expresses around 80% of the target chamber and around 20% of the non-target chamber. So, you know, it's slight heterogeneity in that population, but we have 80% of our target cell there. Now, why do we care about chamber-specific cardiomyocytes? There are chamber-specific pathologies, particularly things like atrial fibrillation, which affects over 33 million people worldwide, and requires a lot more research. You can also have drug-induced atrial fibrillation, so we have a lot of clients looking at those applications for our atrial cardiomyocytes. And there are other diseases, so hypertrophic cardiomyopathy, which will manifest in a specific chamber, so left ventricle. So you might see left ventricular non-compaction or thickening of the left ventricle. So that's why we are particularly interested in looking at chamber-specific cardiomyocytes. So just at a baseline, we see some differences between our atrials and our ventriculars. So here, looking at HD MEA on 3 brains core plate. So this is roughly 4,000 electrodes on a 3.8 millimeter squared chip. Here we see that the atrial cardiomyocytes have a faster field action potential rate. And we also see on the axial maestro system, similar kind of faster field action potential rate here on the right-hand side. On the left-hand side, you'll see that that T-wave notch on the atrial cardiomyocytes occurs a bit earlier. So there's a shorter field action potential duration in the atrials compared to the ventricular cardiomyocytes. With the axion system, you can also convert that extracellular field action potential into a leap waveform, which more closely resembles the intracellular action potential. And again, here you can see that difference quite clearly. So there's a shorter AP duration in the atrial cardiomyocytes. The ventriculars are longer, and they have this kind of more pronounced calcium plateau, which is indicative of ventricular cardiomyocytes. When we look at 2D contractility, we again see similar kind of waveforms. So here at the top, if you just look at the DMSO control, the atrials are a bit more triangular in shape, and the ventriculars show that kind of calcium plateau at the top. But when we interrogate these cells with a cholinergic agonist carbachol, we see dose-dependent effects in the atrial cardiomyocytes and not in the ventricular cardiomyocytes. So we see greater contractile force, reduced beat rate, longer beat duration. And we've tested a number of different compounds on our atrials and ventriculars. And we do see differences despite, as I said, that almost heterogeneity, with 80% atrial cardiomyocytes, 20% ventricular in our preparations. But because of that heterogeneity, what we've now started to look at is even more specific even higher chamber specificity. So we have licensed a left ventricular cardiomyocyte differentiation from Andrea Bernardo's group at Imperial. And essentially, when you look at optical mapping using dia4 ANEPS, you can see with the left ventricular differentiation, you have even more of that pronounced kind of calcium plateau in the AP waveform. The beat rate is slightly reduced, and the triangulation is, the waveform is a lot less triangular. When you quantify these APs and you look at the whole population, we're seeing, roughly 97% ventricular-like APs. So it's much improved from what we've seen before. We also see increased expression of IK1 or KIR2.1. So there's also an improvement in maturity as well. So in AXOL, we've been looking at modulating HAND1 expression. So HAND1 is a transcription factor involved in heart tube looping. So it's an important marker for left ventricle development. So during in-process QC, we're now looking at HAND1 expression, and we've been able to modulate that with BMP4, higher BMP4 concentration to kind of drive that expression. So we're fairly confident now in our chamber specificity and being able to generate these highly specific left ventricular cardiomyocytes. And when we look at ICC, what we see now is complete absence of atrial markers that we used to see in our regular differentiation. So no expression of MLC2A, no expression of ANP. When we look at calcium transient analysis as well, we're seeing faster time to peak, faster time to baseline. So again, it's just that these cells are highly specifically ventricular. We're also seeing some improvements to maturity. Now, speaking of maturity, it's been something of a holy grail in the field for a while now. So many of you will know that iPSC-derived cardiomyocytes are quite immature in nature, and there's always been kind of a bit of a goal to drive even more maturity in these cells. So it can help with things like in vitro drug screening, making that a bit more accurate, disease modeling, maybe seeing even greater phenotypic differences between healthy and diseased, and also aiding in 3D culture, so better structural integrity, contractile force, etc., etc. So there are a number of hallmarks of cardiomyocyte maturity. So I've listed a few here, but things like even their morphology, more mature cardiomyocytes are more rod-shaped, they have more aligned sarcomeres, their metabolism involves fatty acid oxidation rather than glycolysis. Spontaneous beating should reduce, and gene expression of things like KCNJ2 should increase, and reductions in things like HCN4, which are associated with automaticity. So there are a number of ways you can mature cardiomyocytes. So you can culture them in 3D, you can culture them in those nanogrooves, extended culture for 60 days plus, tachypacing, so electrocuting them for a long while. But at AXOL, what we've decided to do is use a metabolic maturation medium to drive the metabolism away from glycolysis to fatty acid oxidation. And what we see in just 7 days of applying this MyoMax maturation medium is more rod-shaped cardiomyocytes, more aligned sarcomeres. We also see connexin 43 expression. So on the right-hand side, you'll see some kind of punctate green dots, and that's connexin 43 gap junctions, so improved cell-to-cell coupling. And here on the right-hand side, this is MEA data that improves cell-to-cell coupling, seems to improve the two-point conduction velocity. The spontaneous beat rate goes down, again, something that we'd expect to see in more mature cardiomyocytes. And on the left, we've got some tempo-seq data, so some transcriptomics, and we see things like calcium handling genes, so phospholamban, ryanodine receptor triadin, all increased after the MyoMax media is added. We see reductions in the expression of things like HCN4 associated with automaticity, and there are metabolic genes, so fatty acid binding protein 3, ACAP1, MDH1, all increased after just 7 days of adding this media. So much more work to do, but in summary, I think it's important to consider donor diversity when looking at cardiomyocytes. And the key thing is that we see differences in drug response between atrial cardiomyocytes and ventricular cardiomyocytes. And so now we're offering left ventricular cardiomyocytes for even more specific chamber specificity, and also driving that maturity, like I say, with the MyoMax maturation medium, which in just 7 days is shifting that metabolism over to fatty acids. So with that, I'll conclude. I think Kadla will take over.
Perfect. Thanks, Jamie. That's great. And you're well within time. So we're going to have hopefully loads of questions rolling in the chat box so that we can be putting them all to the experts at the end. As I say, it's not just the speakers. We've got a third expert, Ataneska Velichkova, because she will be almost our culturing and growth and differentiation expert from Sofian. So yeah, do get your questions coming in. I don't want to take too much more of your time, Kadla. I know you've got some great data to be showing us, so I'll leave it all to you, and I'll come back at the end to do the Q&A hosting.
All right, thank you, Damien. So yeah, my name is Kadla, and I'm a research scientist at Sofian. And today I will present the results of a project where we worked with axoCells to characterize the axoCells using automated PET clamp. So as you know, iPSC stands for induced pluripotent stem cell. They are produced from somatic cells that you can extract from a person and then reprogram into stem cells that you can differentiate and mature into your specialized cell of interest. And today's topic is cardiomyocytes. So we have seen automated PET clamp being involved in the characterization, disease modeling, drug screening, pharmacology, and safety screening of iPSC cardiomyocytes, and we also see potential for personalized medicine. So in this project with axoCells, we aimed to develop assays that would allow automated PET clamp or APC recordings of axoCells ventricular cardiomyocytes for characterization, drug activity, and screening studies. So there are two main requirements if you want to do automated PET clamp recordings of cardiomyocytes. The first one is that you need a mature and homogeneous cardiomyocyte population. As you heard in the first part of the webinar, this is a focus that the cell vendors have and that axoCells has, and they continue to improve their cardiomyocytes. The second requirement is that we need a high quality cell suspension. We need to dissociate the cells into suspension to do automated PET clamp recordings. So cardiomyocytes, they tend to form a very tightly connected tissue-like structure, as you can see here on the micrograph in the left. And if you're not very careful, then they can be quite damaged by dissociating them into the suspension. So we have worked with many different iPSC cardiomyocyte cell lines as Sofian, and we have realized that we cannot find one protocol that serves all, but we have a very good strategy for now in place for optimizing the cardiomyocytes suspension every time we start working with a new cell line. So you can see it outlined here on the left, and we did the same also for the axoCells. There are things you can look at during the culturing stage. You can look at the plating density, the surface treatment, adding different stabilizing enzymes. Then in the dissociations, at the dissociation stage, You can look at the dissociation enzyme used. You can vary the incubation time, look at different centrifugation parameters, and also we see for the cardiomyocytes that they benefit a lot from being replated 24 hours prior to experiments. Then finally, after dissociating them, they also sometimes require further filtering and sorting. So once you have a good cell suspension, and we recommend more than 90% viability and as low aggregation as possible, we can add them to our PET clamp systems. So here on the right, we listed the requirements that the cell requirements that we found with the axoCells. So on our Q-patch, we use 500 to 900,000 cells to cover a full Q-plate with 48 experiment sites. And on our cube, we use 3 to 8 million cells to cover a full plate with 384 experiment sites. And if you need fewer to use fewer cells, you can work with partial plates. So first, I will show some Q-patch data. So on the Q-patch, we can use physiological solutions, which is really advantageous, especially if you want to record cardiomyocytes action potentials. I will come back to that later. So here, I show on the left a Q-patch consumable plate view. This is half a plate, where you can see each site harbors a single cardiomyocyte, and then you see the sodium currents in response to a applied voltage protocol. And with physiological solutions on Q-patch, we get successfully whole cell experiment in about or up to 40% of the experiment sites. So second from the right, you can see the seal resistances. We have put quality seals in the gigaohm range. And on the furthest to the right, you can see a histogram of the C-slow values or the cell capacitances. And in general, for cardiomyocytes, we see that they are larger and more variable, the size of the cardiomyocytes, as compared to stable cell lines. We also see that as the cells mature, the capacitance increases as we would expect. So then I'll just give a couple of examples where we investigate different cardiac ion channels, starting with the TTX-resistant cardiac NAV 1.5 channel. So we see, or we are able to record sodium currents in more than 80% of the cells that we have successfully clamped. In the middle, you can see an example of the NAV 1.5 current in response to a voltage step protocol. In black, it's recorded in saline, and in red, you can see the effect of a blocker, TTX. We can then extract the current and plot it as a function of step voltage, as we have done here on the right. And you can see again in black is the current voltage relationship recorded in saline. In light red, we add 300 nanomolar of TTX, and you can see there's no significant block of the channel as you would expect for the TTX resistant NAV 1.5 channel. And then adding a high 30 micromolar concentration TTX completely blocks the channel. Another example is the cardiac L-type CAV 1.2 channel. So here we can record calcium currents in about 60% of the cells that are successfully clamped. You can see here in the middle, again, an example of the CAV 1.2 current in response to a voltage step protocol. And then we have added different CAV 1.2 channel modulators. In green, the current is blocked by nifedipine, and in red, it is enhanced by the activator BK8644. So on the right, we have plotted again the current voltage relationship, and you see again in black the CAV 1.2 IV curve. Then in green you can see the effect of the blocker, and in red you can see the effect of the potentiator. Then, as I mentioned, because we are able to use physiological solutions on the Q-patch, we can record action potentials. The main challenge when doing that is that iPSC cardiomyocytes tend to have an elevated resting potential as compared to primary cardiomyocytes. That's even when they are patched using manual patch clamp and they are still adhered. And when we dissociate them to record them with automated patch clamp, this problem is even more pronounced. So to accommodate this, we use a feature that we call adaptive current clamp, where we hold adaptively every cell at a current that corresponds to a 90 millivolt resting potential prior to pacing the cells. So using this method, we can measure useful action potentials in also about 60% of the successfully clamped cells. And as you can see here in the middle, there's an example of action potentials from 12 different individual cardiomyocytes recorded within the same plate. We can extract different parameters. So here we have looked at the peak potential in gray, the hyperpolarization potential in blue, the action potential amplitude in red, and the action potential duration in green. And when we plot that together for 52 cardiomyocytes on the right, you can see that the potentials are pretty reproducible, but we do see a quite large variation in the action potential duration from cell to cell, and that very likely relates to the expression of the ion channels that are involved in the formation of the action potential. Then we can also start looking at how compounds affect the action potentials. So here we added the CAV 1.2 channel blocker, nifedipine, and we see, as expected, that the action potentials are shortened as we block the channel, and when we activate this channel, we see that the action potentials are prolonged. So this is just one example of how we can start looking at compound effects on the individual cardiomyocyte action potentials. Then onto the cube. So on the cube, we use seal enhancing solutions, meaning that we can get double the whole cell success rate. So here we are having more than 80% whole cell success rates. On the left here, you can see the cube consumable plate view. So I'm showing 48 out of 384 experiment sites. Each contain a single cardiomyocytes. And again, we are looking at sodium currents in response to a voltage protocol. We are able to record sodium currents in more than 60% of the sites, which makes this really applicable for compound screening. The seal resistances and the capacitances are similar to what we see on the Q-patch, and we can do physical characterization of the sodium channel. Here you see the current voltage relationship on the left, and we can also do activation and inactivation plots and extract V-halves that are in agreement, pretty good agreement with literature values for NAV 1.5. Then we did a proof of concept compound screen using the state-dependent blocker tetracaine. So you can see here the compound layout. On the left, we have the two outermost columns are controls, and then we have increasing concentrations of the compound. We apply a 2-pulse voltage protocol where we adaptively hold the cells at their V-half of activation prior to the second pulse. And then we can plot the normalized currents of the two sodium peaks as a function of compound concentration, as you can see here on the right. And we can extract IC50 values, which are again in good agreement with literature. And we also see the state dependence of the compound, which is much more potent on the second peak. Finally, we also tried exploring a perforated patch clamp as an alternative to the standard whole cell configuration for more physiologically relevant calcium and action potential recordings. Normally, we position the cells on the surface, as you can see here on the left, and then we apply suction to make a hole in the cell and get electrical access to the interior of the cell. But in perforated patch clamp, we instead exchange the intracellular solution during the experiment and add a perforating agent that can then insert into the patch and allow electrical access, but without compromising the integrity of the cell cytosol. So using this technology or this method, we got some preliminary data. We still need to optimize success rates a lot. So the data, we don't have a lot of data, but the data we have do show a tendency towards increased calcium currents in the perforated as compared to the standard whole cell configuration, which also translates into a more prolonged and relevant action potential phenotype recorded in the perforated configuration as compared to the standard whole cell. So in summary, by optimizing the protocols for dissociation of the axoCells, we can now do APC recordings with good success rates. We obtain up to 40% whole cell success rates in physiological solutions on QPatch and more than 80% whole cell success rates in seal enhancing solutions on Cube. The fact that we can use physiological solutions on the QPatch makes this platform very applicable for biophysical and drug activity studies targeting cardiac ion channels and action potentials. And the high throughput nature of Cube make that very relevant for screening of cardiac ion channel modulators. And finally, I showed some preliminary data where we are investigating if we can increase the physiological relevance of calcium and action potential recordings on Cube by using the perforated patch experiments. Then we have a lot of supporting materials if you want to try running axoCell cardiomyocytes on your systems. And you can just reach out if you want to get access to those. And finally, I want to acknowledge the people involved in the project. Atanaska will come on in a minute when we have the Q&A session, and Ayana from Sophion. And on Axel, we worked with Stephen. And then I also want to mention that we recently launched a new product that is highly relevant for cardiac studies, namely this ETKM, which is a platform for high-throughput 3D contractility assays, allowing us now to correlate the ion channel mechanistic studies we can do with APC to tissue function And I want to mention that we have done successful APC recordings from primary and iPSC-derived, many different primary and iPSC-derived cell types and disease models. If you think it's interesting, we are always open for collaborations, and we also have these grants that you can apply for. Yeah, so thank you for your attention, and we will go on to the Q&A session.
Great, Kadla. Thank you very much. That was a really nice talk, and we are well within time, which is lovely too, because it means that lots of the questions that we're coming in, I'm sure we can get round to answering them. For the people that are going to be answering the questions, our experts in the panel, top right, you'll hopefully see a button that will give you all messages or even just questions only. I'm going to be looking at that questions only. So while I'm looking at the questions only and giving these questions to the expert panel, Jetta, if in the background any new questions that come in, they can be added to that questions list and we will go through them as we can. So starting at the first question, which is at the bottom of that list, if you're the speakers trying to sort of follow my questions. Ralph got in very early. So Ralph was asking, and I think this is probably more for Jamie, but by all means, if there's others that think they might be able to add to Jamie's answer. Ralph asks, would you expect sex-specific differences in the properties of the cardiomyocytes if you don't have a medium containing sex-specific hormones?
It's a very good question. I guess just that we don't see sex-specific differences in our cardiomyocytes that we've got on hand. However, I have seen some work in the field. I think it was maybe Laurie Svoboda's group in Michigan. They've got quite an elegant system because they've got iPSC-derived cardiomyocytes from Klinefelter patients. So they've got XX and XY. And so it's like an isogenic pair that they've got, and they see differences in calcium handling between male and female, and differences in response to different toxic compounds. So it's not something that we've seen. I think because we've not got an isogenic pair, we'd need, even more donors just to group them male and female and have a look at something like that. But it's certainly something that we are interested in is, diversity in gender, ethnicity, etc., etc. Not just with the cardiomyocytes. I mean, we seem to see some differences, I think, in the sensory neurons as well. So definitely something that we're actively looking at.
And just to add to that, Jamie, presumably this is all part of the ongoing development developments in these areas, as in you're trying to get them more and more, adult or even patient specific, and that might include, as Ralph's asking, sexual differences, but also maybe age differences, young, middle age, right through to old age, and even geriatric relevant models. So I presume these are all things that people like AXOL are really trying to work towards in terms of getting that, broad range of different models that you would be able to get from iPSC-derived neurons, cardiomyocytes, other tissue types.
Absolutely. I mean, it's something that we're looking at. I think we've termed it clinical trial in a dish in some of our projects that we've done. So, it's something that I think is quite important when you're running these early, drug discovery programs and things.
Yeah. So working our way up the list from the bottom, which is actually the time order they came in. So Saeed came in with a second question. And again, this is potentially for Jamie, but possibly Kadla, even Atanaska may have thoughts on this. Saeed asks, if you decrease HCN4 expression, do you still see autonomous beating in the ventricular cardiomyocytes?
So in our hands, what we see is a markedly reduced spontaneous beat rate, but there are some wells after MyoMax treatment where we see quiescence and we then need to pace cells. So definitely a markedly reduced beat rate. But yeah, we think that's associated with the reduction in HCN4 expression.
Don't know if anyone else on the panel would have anything. Have you tried anything with HCN blockers, Kadla, that might sort of knock out, you know, some of the action potentials you see before knocking out specific, in this case, HCN for ion channel populations?
No, we haven't looked at that. Not yet.
Okay, let's move on to the third question that came in, again from Ralph. Ralph got a few in, so you might have a few to answer to Ralph. I presume it's the same person, but there could be multiple Ralphs asking different questions at different times. Third question from Ralph. Do you have any metabolic data revealing the increased fatty acid metabolism using the MyoMax medium? This is probably more for Jamie again, in terms of how you're developing these different iPSC cell lines.
Yeah, so it's something we're looking at. We've been looking at the seahorse assay, but we're actually probably going to move to the Recipher system so that we can look at it at a more granular level, kind of along a time course. So that's basically, we don't have enough N numbers yet, but it's definitely something we are looking at.
Thanks. So the next question up the list, Shafali asked, and this is quite an interesting one. I'm not quite sure how you do this, which is why it's quite an interesting, potentially even challenging question, but does dissociation affect surface expression of ion channels?
Yeah, so I think the short answer is that it probably affects some ion channels, and we are running different projects to try and assess how much it is affecting and which channels. That I think there's quite a bit of literature showing that the HERC channel, for example, is quite affected by dissociation. I think it is important, and that's also what we recommend anyone that are doing these experiments, that you assess for yourself that the specific ion channel you are looking at and the specific type of experiments that you are doing is not affected by the dissociation, or that you can still do it after having dissociated the cells. In the very best case, if you have access to manual patch clamp, you can compare what is the expression levels when you do your experiments in manual, and then you can see if they are comparable enough that you can investigate the ion channel in the way that you want. But we do see that, for example, the resting potential is more affected in dissociated cells than in when they are attached to the surface. It's more elevated, and that is likely due to either some ion channels being there at lower density, or the loss of the morphology that is then also preventing the ion channels to segregate and position themselves in the right areas of the cell. I hope that answered.
I think so, yeah. And I like your idea of comparing manual versus automated patch clamp, because obviously the manual, you'd be doing it in the adherent on a glass cover slip format. The downside to that, obviously, is you would probably need quite a number of manual patch recordings to get a good overview of a population of cells at time point 24 hours post seeding, or 48 hours, or whatever it would be. You'd probably need, I don't know, a good 15 to 20 manual patch recordings to really give you a snapshot of the population of cells that you're looking at on a glass cover slip, versus the the potentially dozens, if not hundreds, of recordings that you might do on an automated patch clamp. But yeah, I mean, that would certainly be the way to do it.
And it also should be mentioned that there has been a big focus to develop also dissociation enzymes, or different combinations of dissociation enzymes that are suitable for dissociating cardiomyocytes without damaging or with damaging the ion channels as little as possible. So we have 2 or 3 that we usually test and recommend also testing when you do the first tests, just to see how they affect the measurements.
Thank you. Ralph asks, and this is probably for you, Kadla, since you're our expert of how you do these recordings on automated patch lamp. Does physiological solution mean you are fluoride free in the intracellular solution?
Yes.
Good. That
Exactly.
was a nice simple one.
Yeah, and we keep us close to physiological, all the ion compositions as close to physiological as we can.
Yeah, and I don't want to start going into the ultra challenging recordings, but even when it comes to physiology, I know that a number of people that, you know, ourselves really have been pushing this quite a bit, is as well as physiological solutions, we're also trying to do it at physiological body temperatures. So 37 degrees, for instance, for humans, and that's another element of the physiological environment that we're almost trying to do some of these recordings at. So another one, this one specifically for you, and again, might be quite a simple one. For Dr. Rosholm, can these experiments be achieved on your QPatch 2?
Yes, they were done on QPatch 2, the QPatch part of the presentation. So they can. And
then a nice old friend of ours who we've seen a number of times at Ion Channel Modulation Symposium and even in our labs in Copenhagen, Sarif asks, have you seen or used different pH levels for your physiological solutions? What was the exact pH? And thanks for all the nice talks, everyone.
I think the pH is 7.4, and we haven't used different pHs for the experiments. But if you have something you want to investigate, you can let us know.
Because I mean, almost expanding a little bit on what Sarif might be asking. For instance, in cardiac ischemia, I believe that the pH does become more acidic. So, you know, if you were to potentially model that pathophysiological environment, you know, we were just talking about environmental conditions and trying to keep them physiological. There are obviously some disease states that would be, you know, changed pH, for instance. I think cardiac ischemia might be one of them. So, you know, that could be something where to try and keep the pathophysiological environmental conditions more relevant, you might go to more acidic pHs.
Yeah, sure.
And then moving on, so Ralph asked, did you try to record and activate late sodium current? Again, that's probably for you, Kadla, arguably.
Yeah, we didn't do it in this study, but it could be something that we could look into. But no, we didn't do it with the axoCells.
Okay, moving on to the next question, moving up the list. YE asks, do you have success in recording potassium currents in iPSC cardiomyocytes such as hERG channels, KV7.1, KCNQ1, and KATP? And then the second part of that question, if yes, can you comment on the batch to batch variability of potassium current recordings? Probably for you, Kadla, but maybe Atanaska might have something along those lines too. I don't know.
At least I can start. So with the axoCells, we haven't done these experiments yet, but we have with other iPSC cardiomyocyte cell lines recorded hERG and the IK1 current. So IKR and IK1, they are the ones that are notoriously difficult to record in iPSC cardiomyocytes, but I think also as the cardiomyocytes are getting better and better and more mature, hopefully it gets easier. It is still tricky, and we have developed some assays where we enhance the currents and then we look at the sensitive currents, adding some specific blockers and so on to be able to detect it. Yes, we can look at the potassium currents, but it is still tricky. We haven't specifically looked for HERC in the axoCells, but it is on the drawing board as something we would like to do. So I don't know, Jimmy, if you can comment on the potassium currents in your cells.
So I guess the IK1 is always challenging in iPSC-derived cardiomyocytes. So again, that kind of informs why we wanted to pursue the MyoMax maturation media to kind of boost expression of that. In terms of HERG channels, so we've super-validated our cardiomyocytes, our ventricular cardiomyocytes, so we see sensitivity to HERG block and prolongation of APD. So yeah, it's definitely there. It can definitely be used for that kind of cardiac safety assessment when you're looking at HERG. But definitely it would be good to do some automated patch for the potassium as well. I think it'd be a good data set.
Moving on to a second question from YE. What is the current success rate in using perforated patch configuration?
That is a good question. Atanaska, do you recall? I would say 10%.
Yeah, I think it's 10%. It's been very challenging to establish the perforated patch, and I think currently we are at 10-15%.
Yeah, we can.
Sorry, Kadla. What's the reason for that? Is that partly due to? I mean, perforated patch is that bit more challenging anyway. You've got to make sure you're obviously getting the perforations correctly to get the electrical access. But do you have a reason, or do you have any thoughts on why that success rate might be quite a bit lower than standard wholesale recordings?
Yeah, so, and Atanaska, you can just also break in if you have more to say, but I think in this, for the axoCell, we have worked with different cardiomyocytes preps, and it's always difficult to do the perforation, and it's always difficult to get the success rates up. With the axoCell specifically, the challenge was that you need high enough suction to position the cells on the patch hole, but it should not go spontaneously in whole cell. And we actually, because the cells are a bit larger, it require quite a hard suction. And what we see is then, because the cells maybe are a bit fragile, then they go spontaneously in whole cells throughout the experiment, and then you cannot use the experiment. So that is one thing that is really tricky. So you need to find a balance where you are both capturing a good portion of cells, but they are also stable throughout the experiment in the cell test configuration. That was one part of it, and the other part of it that turned out to be challenging was also to find the correct concentration of the perforating agent. So we tried optimizing this, but to be honest, it's not finalized. It needs further optimization to get a better success rate.
Thank you. Erica, on perforated patch, also adds, which perforating agent are you using to achieve perforated patch configuration recordings?
We use nystatin. We have tested with stable cell lines, amphotericin. and beta-acene and nistacene, and then I think I seem to recall there's one more, but nystatin works best in our hands.
And often with these perforating agents, it can be quite cell line specific, almost as you were saying, in terms of figuring out the concentration and the timings and the pressures. There's a number of things you almost need to optimize for, and part of that could even be for cell line X, we use perforating agent A, and for Y, cell line Y, we use perforating agent B. It can even be down to cell line specificity of which one works best.
Yeah, and also which ions it allows to pass through and so on. Yeah.
Okay, moving on. Ashley asks, do you think that iPSC-derived cardiomyocytes are best used for safety assessment or for drug discovery? Probably a bit of both, to be honest, but who wants to take that? Jamie?
Yeah, I can start. It's a bit like choosing between which one of your kids is your favorite, isn't it? I guess on balance, maybe cardiac safety assessment, particularly the ventricular cardiomyocytes. As I said before, we SIPR validated our cardiomyocytes for herd liability, and we can use the ventriculars to predict herd block. So I guess in that sense, the safety assessment. But for drug discovery, if you take the atrial cardiomyocytes, we've got a model now where we can tachypaste them and essentially see a phenotype that resembles atrial fibrillation, and then we can then use that to look at to rescue that phenotype. So it's a little bit of both, but I'd probably go more on the safety assessment side of things.
Okay. And Schnitzana asks, thank you for interesting presentations. Have you had a chance to use MyoMax for disease modeling like LQT, CPVT, or SQT? If yes, what are the main parameter differences between standard and MyoMax media? Probably for you, Jamie, I would guess. Yeah,
so none of those diseases that are mentioned, but what we've got at axoCell is we've got a family cohort of Friedreich's ataxia lines. So with Friedreich's ataxia, there's mitochondrial disruption. So we're working on looking at that and then using the MyoMax to see if we can kind of increase that phenotype that we're seeing. So yeah, it's that disease, it's Friedreich's ataxia that we're really focused on to kind of boost the phenotype with the MyoMax media.
Thank you. Saeed asks, in your maturated cardiomyocytes, did you see expression of inward potassium current? It might be outward current, but irrelevant. A potassium current, were you able to see them on the Q-patch cube? Presumably that's you, Kadla, and or Atanaska?
Yeah, so we, as I mentioned before, we didn't look for them yet in the axoCells, so I cannot say for those, but we have measured them on Q-patch previously for other iPSC cardiomyocytes. But it is, as I also mentioned before, the inward one is a tricky one for iPSCs, and we only see them in some of the cell lines that we've worked with.
And I realize that we are getting close to the end of the hour's webinar, but I'm available, and if the expert panel are still available, stick around, and we still seem to have quite a number of people who have logged on and online. So we will keep going as long as we can, or until the panel start falling asleep or needing to dash off to look after kids or whatever it might be. But we'll do what we can to get through as many of these remaining questions, because we've got so many that have come in already. We've answered quite a few, but we've still got a few to answer. So I'll crack on with what we can, and if you do need to dash off, just dash off and, whether that be the audience or even the expert panel, I might be here on my own and answering the last few questions randomly. Don't let me do that. Okay, what is so Saeed asks again, what is the success rate without fluoride?
Yeah, so we ran without fluoride on QPATH, and then we obtained with the axoCells up to 40% success rates. The success rate vary from cardiomyocyte cell line to cardiomyocyte cell line, and we have, I think, reached up to 60% success rates in physiological solutions for some cell lines.
Ashley asks, which Sophian platform is best or most commonly used for safety versus drug discovery, and then the reverse of drug discovery versus safety, if that makes sense? Probably you, Kadla and or Atanaska.
Yeah, I'm not well, I think both Cube and QPATH are used with standard cell lines for safety and drug discovery. There, of course, it depends on your throughput requirements. The Cube is high throughput, and if you are testing library of compounds, whether it's safety or whether it's drug discovery, then you would go for that one. When you're looking more into fewer compounds or maybe mode of action studies, you would maybe tend to use the QPATH. For the cardiomyocytes specifically, it's you have the throughput on Cube, but you have the possibility to work with physiological solutions on QPATH. So it depends on what you want to investigate. So for example, if you want to, as it is right now, if you want to look at cardiac action potentials, then QPATH is the best. But if you want to screen sodium currents, then Cube would be the preferred platform. I'm not sure I completely answered the question, but I hope it was understandable.
I think that's a pretty good answer. You almost choose the tool depending on what your key sort of aims are in achieving it. If it's really huge numbers of compounds, the higher throughput will be your friend. If it's greater mechanistic detail or physiological conditions, then and with smaller numbers of compounds, then QPATH is probably a slightly preferable platform. Yes, it really depends on what questions you're trying to answer. The platform might be most suitable for. I think Sarifa was just confirming that yes, cardiac ischemia is potentially one of those conditions where the pH might be more acidic. So going on to Brian's question, how long does an average iPSC cardiomyocyte last in both the QPATH and Cube assays? Good question, Brian.
Yeah, I would say it's about an hour. The experiments are typically about an hour. I don't know, Atanaska, if that also goes for the Cube assay.
Yes, I think it was about an hour with the perforated patch. There was a bit more also waiting time. So I think an hour and a half was probably the maximum goal.
And just to add to that, that might sound quite long, Brian, if you're used to manual patch. And what we find is with planar patch, effectively where you've got recording sites in planar arrays in a recording chip style format, as you would do on an automated patch clamp, that form of recording is far more stable in terms of the seal, in terms of the duration of your seal, the quality of the seal over longer periods. because rather than having a huge electrode that acts effectively as a mechanical antenna, you know, picking up, even with a very good air table, your large electrode that you have on your cell in a manual patch format means that that electrode tends to pick up mechanical noise far more readily than a planar array of recording sites. So 60 minutes to 90 minutes on an automated patch clamp platform using that planar recording site versus the manual patch electrode version, they can be really quite a bit longer. So one thing to keep in mind in terms of throughput isn't just the number of cell recordings you're making, but actually duration as well. Because if you're getting 20 recordings that are an hour plus in duration, you've got 1200 minutes of recording experimental time. That versus what you might achieve manually, where arguably a 10 to 15 minute or maybe a 20-minute recording might be a good recording manually because of that additional mechanical noise that the electrode seems to bring into a manual patch recording system. It really makes quite a big difference in terms of the amount of experiments that you can achieve, you know, different compounds that you could be applying, for instance, in a 60 to a 90-minute recording versus a 10 to 20-minute recording manually. Moving on, because we do still have more questions, so I need to keep my responses and answers shorter too. Ashley asks, where do you see the EthicA-M fitting into drug discovery versus safety? For example, cost versus time to generate the tissues versus single cell based assays.
I guess one thing I like about the EthicA-M system is, you know, when you make these 3D, you do get kind of more, well, first of all, it's a genuine contractility response. It's not an image-based kind of pseudo contractility. And what we've been seeing in these 3D HTs is you get more mature responses, so positive force frequency, dose-dependent positive inotropy with, you know, things like isoprenoline. So yeah, and I think the price of things is it's coming down. I think the EthicA-M, it's something like 40,000 cardiomyocytes per construct, something like that. So it's, you know, from a vial of cells, I think you can get a number of tissues to kind of help you get higher throughput.
Would you have anything to add to that, Kadla or Atanaska?
Yeah, no, the only thing I was seeing in how it's fitting into drug discovery is what we see is EthicA-M is a complement to like a human assay that is more complementary to, for example, animal studies and might identify drugs or cardiac risks in a more human-like setup as compared to the animal studies, and it might also have the potential to reduce the amount of animal studies required. So it's at that level, whereas I see, and I'm a bit out of my comfort zone here, but as I see the APC and the initial screening coming before, and then you would move on to the contractility assays when you have identified, when you have done the first screen of compounds.
I'm aware of the time again. We're now obviously sort of 8-9 minutes past the hour, but again, I'm okay to stick around. We've still got quite a number of people logged on. So if the panel are still okay going forward, then we can carry on and try and get through a few more questions, try and get through as many as we can. Okay, so I've got to try and figure out where I am on the list. Yeah, so Shafali asks, is there a difference in sodium, calcium and potassium channel currents in potassium fluoride versus potassium fluoride solutions?
Yeah, so the calcium currents can be affected since calcium fluoride precipitates. So that is when we have challenges with potassium fluoride solutions, then it's typically related to the calcium currents and the calcium equilibria in the cells being disturbed.
I think we might be down to the last two, so I was probably preempting that idea of the panel having to sort of take off and pick kids up, etc. I mean, if you do need to obviously go, but we'll try and get through these last two. Ralph asks, have you ever tried to culture cells or even cardiomyocytes on the whole of Q patch plates and to get access to them? That's a really nice idea, Ralph. I wonder if we've thought about that. Go on, Kadla.
We are looking into that option. We haven't tried with cardiomyocytes, but we have for neurons. We have had a bit of focus on trying to make this feasible. So yes, it's something that we are looking into. We don't have any meaningful results to show you yet.
And the final question, so no pressure, Hugh, you've got the last question to the expert panel, so this better be a good one, Hugh. What is the magnitude of the upstroke velocities of the action potentials when using adaptive current clamp? And in Q versus Q patch, is there a difference?
That is a good question, and this is something that we can extract using our analysis software, but we haven't looked at it. Of course, during the pacing in the first part, you cannot really analyze it because you are changing due to the pacing. But then we often see that the cell takes over, and you can see the sodium channels opening, and therefore the upstroke of the action potential. And in that part, I think we could quantify it and have a look. But we haven't done it, so I cannot really answer with a number. Yeah, and regarding the adaptive current clamp, it is true that this that you are holding the cell at a current to induce a more relevant membrane potential, of course, comes with the cost of some of the ion channels being affected by this. Also, after pacing, we are reverting back to this adaptive current, and that also affects the ion channel function. So it's a correct observation that this is something that you need to be aware when you do the experiments, and be sure that you're not artificially changing something that are affecting your results.
Great, thanks everyone. Thanks to our speakers, Dr. Rosholm and Dr. Bhagwan. Thanks to Atanaska for your input on the Q&A at the end there. We obviously had quite a number of questions and some really good, answers and, discussions around it. So thanks to all the panel, but thanks also to you, the audience, for sticking around so long. We've still got quite a number of you still logged on, so thanks for sticking around right till almost 4:15. And we will hopefully see some of you, if not all of you, again at the next webinar in probably a couple of months' time. I hope we will be seeing you then, and till then, take care. Happy research, and go on with your brilliant stuff that you're all doing out there, and we will be presenting our brilliant science to you in the next webinar in the coming months. Thanks again to everyone, and take care. Bye then.